Perspective. Today video presentation, my topic is about whole genome sequencing and phylogenetic analysis of human metapneumovirus strain from Kenya and Zambia. Next slide is I will start my presentation with introduction. So what is human metapneumovirus? So HMPV is a single-stranded RNA virus and can cause acute respiratory illness in young children. So other characteristic is HMPV genome consists of 30 kb in size and consists of 8 operating frame. So most of this study is conducted in Africa. In this study, we have 3 methods. So I will start with first method. The first method is HMPV detection and genotype assignment. So nasopharyngeal and oropharyngeal swab sample were collected from children aged 1 until 59 months old hospitalized with pneumonia. So this sample we get from Kilifi County Hospital, Kenya. So the sample were tested for HMPV by RT-PCR assay. Second method is whole genome sequencing. So there are 4 steps of whole genome sequencing, which is primer design, preparation of primer mix, cDNA and PCR, and last step is Illumina sequencing and data analysis. Last method is phylogenetic analysis. So this dataset consists of 138 sequence entries. Next, we start with results. So the first result is genome characteristics. So this table here show a sequencing result and data assembly metric for the 5 HMPV genome. The second result is sequence diversity and phylogenetic relationship. So this table here show the level of nucleotide and amino acid identity of sequence within the 2 major HMPV groups. So the 2 major HMPV group is one from the new sequence of Kenya and Zambia, and the other group is global sequence retrieved from gene bank. The third result is sequence diversity at RT-PCR target region. So the whole genome sequencing was successful for all 5 clinical samples. Table 1 shows sequencing and data assembly parameter. The overall nucleotide identity between all 143 genome sequence analysis was 58.2%. Next is discussion. So in this discussion is HMPV genome sequence data from Africa is very few and information on genome wide diversity is limited. So the whole genome sequence of 5 HMPV strands from Kenya and Zambia were determined and compared. So in recent years, HMPV has been detected at low predominance in Kilifi. The last is conclusion. So in this study, we get a first whole genome sequence of circulating HMPV strain from sub-Saharan Africa. So there are limitations of sequencing method occur in this study. So the result demonstrate the application of amplicon sequencing to generate full-length HMPV genome directly from clinical sample. So one of the advantage of whole genome sequencing of HMPV is for design of local molecular diagnostic. So there are also method and data that can be a reference for studies aimed at understanding HMPV epidemiology and evolution in Africa.